Hello everyone, I'm Pfleger, and this is going to be a presentation about my research group and some of the things we're interested in studying. If you want more information, please visit our website. You can either find it from the address here on the screen or from the department's register of all the faculty web pages. My email is my name at wisc.edu, so feel free to contact me at any time. So a little bit more about me. So I grew up on the East Coast, not a Wisconsin native, unlike many of my colleagues, but I've grown to love many of the things about Madison and about the surrounding Wisconsin countryside. You can see these from some of the hiking trips I've taken with my family and some of the time I've spent outdoors on the ice enjoying one of my favorite pastimes, and that's the sport of hockey. So my chemical engineering career started at Cornell University, where Mike Duncan introduced me to the entire concept in orientation. I fell in love with the idea, pursued it for 4 years there, really enjoyed my experiences. As part of that, I spent a co-op semester with 3M out in the Twin Cities for a summer. Learned I loved everything about research, hated everything to do with material science. So from that point on, I went out to the University of California, Berkeley, and spent 5 years studying with Dr. Jay Kiesling, learning about synthetic biology and metabolic engineering, things that I continue to study these days. And then spent 2 years at the University of Michigan with David Sherman in his group, where we learned how natural products, a lot of the things that become antibiotics and different types of pharmaceuticals, are produced and how they're synthesized by microorganisms. So as you can imagine, I have lots of different interests. I practice what I preach. I do fermentations at home, producing our own fermented beverages. So this is the team I have the privilege of working with every day. And so you can see I've got a large group, about 15 scientists at the moment. We have a new postdoc, Audrey Yanez Gutierrez, who's going to join us here in March. And then sadly, Taylor Cook is about to defend his PhD thesis here in the spring as well. And so we'll be saying goodbye to Taylor and hello to Audrey. But you can imagine we have lots of people coming and going, coming from all parts of the world. Primarily at the moment, we're chemical engineering dominated. So most students have their bachelor's degrees in chemistry or chemical engineering. But from time to time, we will get PhD students who are pursuing microbiology degrees as well. And so we've had a long history of that, particularly in our cyanobacterial research projects. So when you think about our research, it's hard to kind of categorize everything into a small set of words. And so what I did was created this word cloud from all of the papers that we've written over the years. And I was really surprised by some of the things because what I didn't find really was the phrases applied microbiology and synthetic biology. And those are the topics that I think best characterize the work that we do. We genetically engineer microbes and then use them in technologies for the betterment of society.
So to better describe our work, I'm going to use this set of chemical reactions that comprise ways to go from glucose or glycerol, things that cells can eat, to a molecule that's of industrial importance, and that's 1-octanol. 1-octanol gets used in a lot of different applications, but primarily is the precursor to 1-octene that gets used in lots of polymer products. So in microorganisms, you can make fatty alcohols like 1-octanol from fatty aldehydes, and these can be derived from different types of organic fatty acids or acyl-CoA or acyl-ACP thioesters. And so the way that we would think about projects in this area would be to say, okay, which of these reactions can be catalyzed by a given organism that we want to work with, which have enzymes that need to be brought from other places, so maybe other organisms or designed de novo. Do those enzymes have the correct properties, the catalytic rates, the stabilities, the ability to be expressed in high numbers? And can those enzymes be combined without hurting the cell's physiology in other ways? And so if you can do all of those things, you may need to discover different types of proteins. And if they've never been worked with in organisms before, you may need to discover how pathways are regulated and how you can connect new enzymes to those pathways. You'll need tools for developing ways to turn the DNA that you would put in as the new blueprint into the proteins that you use in the metabolism. And then we spend a lot of time with classic metabolic engineering. And this is just like chemical kinetics. We're looking for the rate limiting steps and then taking genetic interventions to try to bypass or improve the rates of those rate limiting steps. Now, we're pretty good at what we do, but I would say the vast majority of our initial designs fail miserably. And so we spend a lot of time troubleshooting. And so we do that by looking at system-wide measurements of things like RNAs, proteins, metabolites, and other things that we use then the techniques of systems biology to help analyze. And then lastly, we like to take some of our things and put them into actual bioprocess situations to see how they would perform, and then potentially file patents that companies can then license and use to use our technologies to make actual products. Now, I can't say that our work is all just in our group. We have lots and lots of collaborators. So within the department, we have active projects, including co-advising situations with the folks that you see circled here in green. We also have relationships with 2 Department of Energy bioenergy centers. So the Great Lakes Bioenergy Research Center is the one here on the UW-Madison campus. We work with a lot of folks within that who are both subject matter experts as well as people who provide access to techniques and instrumentation. One example would be Michael Thomas, who we work with in trying to convert waste materials at the end of fermentations into something valuable. So that way we're getting value from every molecule in the biomass that we feed to these processes. In addition, Professor Thomas and I have a project through the National Science Foundation to study the production of natural products, so things that become antibiotics and other molecules. Similarly, we have a relationship with the CABI, the Center for Advanced Bioenergy and Bioproducts Innovation. This is a center that's primarily out of the University of Illinois, Urbana-Champaign, but has collaborators also at Penn State University in Nebraska and several other universities. And so we work with Costas Moranis, who provides a systems biology perspective that we used to get locally with the late Professor Jennifer Reed's group. But Costas and I have had a long-standing collaboration and relationship on other types of projects, including one through the National Science Foundation to engineer proteins using optimization tools through structures that you've captured in crystal structure studies. We have other projects that we collaborate with a group at Vanderbilt. This is primarily in the area of cyanobacteria. And so Jamie Young is an expert in flux balance analysis. And so this is or metabolic flux analysis, I should say. And so this is where we use labeled carbon molecules to actually measure the flow of carbon through metabolism. And so we use those techniques to help improve the cyanobacteria's performance in producing certain types of specialty molecules. And then there's lots of projects that we're working on currently that are still in the proposal stage. So Professor Zavala and I are working with other groups around campus to look at phosphate removal from manure wastes. And this is a project we're going to submit to the NSF during the recruiting period. And we're working with a company called LanzaTech on technologies to produce chemicals in a very carbon efficient way using microorganisms. And so these are all projects that you might have the chance to work on next year if they end up being funded. I wanted to close with a couple of vignettes. So maybe we'll dive into a little bit more about the techniques that we use. And this is a story that was put together by 2 graduate students who recently moved on to industrial careers. So Jackie Rand was a graduate in 2017. She's now basically a protein production scientist at a local manufacturing company called Aldevron that makes things such as Cas9, the protein that people use in genome engineering techniques. And Chris Mayer, who graduated 2 years later, now works for Genomatica as a fermentation engineer. And so the project that they worked on was focused around this molecule here circled in blue called libulinic acid. It's a molecule that the Domestic Group and many others have shown can be made very efficiently from biomass. And so we thought it might be a molecule that could be accessible for fermentation use that bypasses many of the challenges associated with converting biomass into fermentable sugars. The problem was, at the time when Jackie started this project, we didn't know metabolic pathways or the enzymes that would allow for cells to consume this particular substrate. And so she combined a whole series of genetic, functional genomics, in vitro reconstitution, where she purified all of these enzymes, and then complicated mass spectrometry experiments because many of these molecules were CoA thioesters. So she had to do the experiments using mass spec as the detection method. But through that combination of experiments, she was able to elucidate all of the roles for these 5 enzymes. And this was a study that she published in Nature Microbiology right before she graduated. And so at that time, Chris was very interested in this pathway because he had seen that you could take this set of enzymes, put it into E. coli, and once it was adaptively evolved, those cells could now grow on libulinic acid and produce the propionyl-CoA and acetyl-CoA central metabolites that would allow the cell to grow. And in the course of that study, he discovered that cells also produced a little bit of this molecule called 2-butanone, which you might also know as methyl ethyl ketone. Of course, this is a really valuable molecule. And so what he decided to do was enter a metabolic engineering study to try to produce more of this 2-butanone from levulinic acid. So to do that, he overexpressed an enzyme that provides the decarboxylation chemistry necessary to convert 3-ketovalerate to 2-butanone. And that 3-ketovalerate comes from a product in the end of the pathway that Jackie had discovered. And so in this instance, there's a transferase that takes the coenzyme A, moiety, and transfers it from this intermediate called 3-ketovaleryl-CoA to acetate to produce acetyl-CoA. And then that provides, the transfer then removes the CoA from the 3-ketovalerate. And so when Chris was working on this, he said, I'm going to need a supply of acetate and a supply of levulinic acid to produce our methyl ethyl ketone. And so he built models in the computer to look at this. And this was a collaboration with Jennifer Reed's group. And we looked at the potential butanone production levels as a function of the cell growth. And what he found was there was a normal triangular shape like this, which basically said that if cells were growing at their maximum rate, which remember is the evolutionary objective, then they wouldn't be producing much of the butanone. And if they wanted to maximize the butanone production, they couldn't be putting any of this carbon into biomass. And so this is what we would call an uncoupled situation. And so when Chris took a look at this set of cells, you can see the blue curves here are the levulinic acid, and the acetate is in gray. And so when these were fed to cells at the same time, the acetate ended up being consumed first, even though it was in excess. And so once it was gone, the processing of levulinic acid stopped because there was no ability to do this last step, and cells couldn't get from the acetyl-CoA back to acetate. Now, instead, if you take and delete certain genes, so you prevent the cells from being able to produce the acetyl-CoA and propionyl-CoA directly, and you block other methods of converting acetate into acetyl-CoA, now cells have to send flux through this particular reaction catalyzed by 8-ODA in order to produce the acetyl-CoA they need to gain energy. And so in that instance, when you model it, you'll see that at the maximum growth rate, right, which is on the x-axis, it's at a point where the cell has to be producing a maximum amount of butanone. And so when we implemented this, or when Chris implemented this, I should say, you'll see that the two curves for acetate and levulinic acid are co-parallel, and the acetate doesn't end up being completely consumed because it can't be used without processing levulinic acid. And so what this allowed us to do then was have a unit conversion, bio conversion that processed all of the levulinic acid very efficiently. And so this was a project that he published in Metabolic Engineering in 2019. So I've mentioned a lot about the funding agencies. We have projects that are supported by the Department of Energy, the National Science Foundation, the USDA. There are many fellowship programs on the UW campus that have supported students. So these are the biotechnology training program, the genomic sciences training program, and the chemical biology interface training program. Oftentimes students ask me where my researchers end up going afterwards, and I can't really give you one destination. So this is a set of companies and places that, or institutions that my students and postdocs have gone on to work. So there are places that like Max Planck, where we've got a postdoctoral scientist right now, the Novo Nordisk Foundation Center at DTU, another set of postdocs. We have several people who are postdocs here at UW in different labs. We have folks who have gone on to Boston Consulting Group. Many of our recent cyanobacterial workers our students have gone on to Benson Hill Biosystems, which is applying synthetic biology in plant systems, which is fantastic. Jeff Cameron went to the University of Colorado Boulder as a postdoc or as a professor. And then Andrew Markley, another postdoc in the lab, took his ambitions and created his own company. And so now he sells a product called hop tea, which is a tea that you brew in the presence of hops, and it's a carbonated beverage that is quite delicious, and you can pick some up at Whole Foods if you're actually interested. So that's where my students go. If you have other questions, I encourage you to visit our website or email me. I'm happy to answer any of your questions. And so thank you for your time, and I look forward to hearing from you.